Hello and welcome to all! Thank you for joining us for the first pitch talk session of the day. At the end of the session, please follow the link to vote for your favorite presentation. Now please welcome Morgan Perard from Université Laval.
Hello everyone, I am Morgan Berra, PhD student in Westlatte Laboratory at Quebec City, and I am working on Parkinson's disease. I don't think I am wrong if I said that everyone would like to have a power, teleport, fly, control fire or ice. In my laboratory, our power is to control alpha-synuclein aggregation directly inside neurons. We can start this aggregation where and when we want, as you can see on the bottom left with the little red dot in the cells. But why this power is so interesting? Even if alpha-synuclein is a little protein, it seems play a key role in the pathogenesis of Parkinson's disease. To understand this role is very important to develop efficiency therapeutic strategy. Currently, this strategy against alpha-synuclein are similar as what we can learn in the school with the fire triangle. But here, the triangle is composed to synthesis, clearance, and aggregation. For my PhD project, I am more focused on aggregation process. This process is like a snowball effect. There is a little snowball at the top of the mountain, represents the first pathological seed of alpha-synuclein, which when it descends the slope, the ball accumulates more snow, so more alpha-synuclein, getting bigger and impact the nerve cells. With our power, we will be able to obtain important information about this process and develop efficiency strategy against alpha-synuclein to stop or to slow down alpha-synuclein aggregation, to prevent the formation of the little snowball at the top of the mountain, or to block to getting bigger when it descends the slope. So control alpha-synuclein is a great power. And if you want to know more about this power, I recommend you to follow the oral presentation of Razanshita just after. Thank you for your attention.
Thank you, Morgan. Next, please welcome Reda Fazazi from Université Laval.
Hi, my name is Reda Fazazi, and I will talk to you about my thesis on multiple sclerosis in the next 3 minutes. Canada is one of the most affected countries in the world by multiple sclerosis, or MS. 1 in 380 people is affected by this disease. Unfortunately, this means that someone you know will likely be diagnosed with this condition sometime in their lifetime. But what is MS? Multiple sclerosis is an autoimmune neurodegenerative disease. What this means is that the immune cells, which are kind of little soldiers that usually fight viruses and bacteria in your body, have a recognition problem and will recognize the layer that covers the neurons as an invader and attacks them. Unfortunately, this leads to the breakdown of neurons, which will cause nervous problems. Indeed, these symptoms will start with visual disturbance, incontinence, disturbance of balance, but can quickly worsen and result in serious motor problems up to paralysis. My research team is interested in the well-known sex differences in MS. Indeed, women are 3 times more affected than men by this disease. However, men progress much faster and more severely than women. Several studies have therefore focused on the role of hormones to try to better understand these differences. Despite some success, several questions unfortunately remain unanswered. My research team is therefore interested in understanding this phenomenon of difference in severity between the sexes, but in another way. Indeed, we have identified that the sex chromosomes, which determine the biological sex of an individual, have an important role to play, and that one gene in particular, JARID1C, would be related to this difference. My research projects therefore consist in characterizing and trying to understand more the link between this gene and the severity of MS. Brief topo of cell biology, each gene code for a protein and a protein fulfills a very specific function in a cell. What we have found is that in immune cells, the JAD1C protein is present in much higher amounts in women than in men. Additionally, we have identified that the amount of JAD1C correlates with the severity of the damage that immune cells cause to neurons in MS patients. Thus, men immune cells cause much greater damage to neurons than women immune cells. This could partially explain the difference in the severity of the disease, since the more neurons are damaged, the more severe the symptom of the disease. Our research will therefore, I hope, lead to a better understanding of the differences between the sexes in MS and could open the door to developing treatments that are more adapted and personalized according to the biological sex of a patient.
Thanks, Reda, for sharing your work. Next, we have Sarah Leping from McGill University.
Do you remember the Ice Bucket Challenge? This was a viral trend that involved pouring a bucket of water over a person's head to promote awareness of the disease amyotrophic lateral sclerosis, or ALS. Hello everyone, my name is Sarah Lepin and I am an MD PhD student at McGill University. And it is my pleasure today to tell you about my PhD project on modeling ALS using stem cell-derived motor neurons. ALS is a neurodegenerative disorder characterized by the loss of motor neuron. This causes a progressive paralysis that's usually fatal 2 to 4 years after the onset of disease. Despite extensive research efforts, there are only 2 treatments approved with limited benefits, and the mainstay of treatment is mostly supportive. This slow translational progress of ALS research could be explained by the lack of physiologically relevant model and our incomplete understanding of disease pathogenesis. My goal is to use motor neurons derived from stem cells, which express ALS-causing mutations, to increase our understanding of this disease. In the last decades, induced pluripotent stem cells, or iPSCs, have become a powerful tool to study disorders like ALS in human, disease-relevant cells. Briefly, iPSCs can be generated from somatic cells of a patient or healthy individual back to a pluripotent state, where they have the potential of differentiating into any cell type of interest. Using CRISPR-Cas9, we edited a control iPSC line and generated 2 knock-in lines expressing ALS-causing mutations in the gene TARDBP. TARDBP encodes TDP43, which is the main component of aggregates, which is the pathological hallmark of this disease. Successful introduction of the mutation was confirmed by sequencing, as well as digital droplet PCR. We also confirmed that both lines express the pluripotency markers, including NADOG and TRI-160, and display a normal karyotype. From there, our group is able to reproducibly differentiate iPSCs into motor neurons. Briefly, cells are first differentiated into motor neuron progenitor cells, or MNPCs, which express the markers PAC6, nestin, and also OLIG2. MNPCs can be expanded, banked, or further differentiate into motor neurons, which express islet-1 and the cholinergic marker vesicular acetylcholine transporter. So using qPCR and immunofluorescence, we confirm that TARDBP mutations do not impair normal differentiations into motor neurons. And we will use this model to dissect the molecular and cellular effects of these mutations in motor neurons, which we hope will aid in identifying new therapeutic targets for ALS. Thank you very much for your attention.
Thanks for presenting with us, Sarah. Next, please welcome Laura Martinez Judis from Université Laval.
Hello, my name is Laura Martinez, and now I want you to imagine forgetting your loved ones, or forgetting what your home address is, or even where you place your belongings. These are some of the main symptoms that people think of when they think about Alzheimer's disease. In fact, Alzheimer's disease is a progressive and irreversible neurodegenerative disorder that affects more than 50 million people all over the world. And in fact, Alzheimer's disease patients show progressive memory loss and also continuous worsening of their thinking and social skills, all due to the irreversible death of brain cells called neurons. And these alterations are caused in part by the accumulation of 2 proteins, amyloid beta or A beta and tau, that form into harmful clumps. There are other proteins present in neurons called kinases that are able to tag at specific locations tau and the precursor of A beta through a process known as phosphorylation. And in Alzheimer's disease, the phosphorylation of the precursor of A beta will increase the production of A beta and its accumulation into clumps called amyloid plaques, while the excess of phosphorylation of tau will lead to its accumulation into also harmful clumps known as neurofibrillary tangles. And in this context, our laboratory is interested in a kinase named pol-like kinase 2 or PLK2 that is highly elevated in the brains of Alzheimer's disease patients. So we believe that the abnormal accumulation and activity of PLK2 will increase A beta and tau accumulation. And thus that using drugs capable of blocking the phosphorylation capacity of PLK2, we could reduce the neuronal dysfunction and death associated with these events. So to test such an approach, we decided to use transgenic mice modeling Alzheimer's disease that we treat with a specific drug at early stages of the disease. And we then compare the effect of the treatment to non-treated Alzheimer's disease mice. And what we expect is that this drug will reduce memory loss that we measure with different behavioral tests by reducing the abnormal modification that induces or causes A beta and tau to form into toxic clumps. So in summary, my PhD project, which is carried out under the supervision of Dr. Abidou Eslati and Dr. Serge Rivet, targets the blockage of PLK2 as a new potential treatment to slow down the progression of Alzheimer's disease and related disorders. And if you want to know more about this, just come visit my poster later on. Thank you very much for your attention.
Thank you, Laura, for presenting your work. Finally, please welcome Maxime Teixeira from Université Laval.
Every day, 25 people are diagnosed with Parkinson's disease. So between 2011 and 2031, the number of Canadians affected is expected to double to more than 150,000. Parkinson's disease is the second most common neurodegenerative disorder in the world and is characterized by the selective loss of the dopaminergic neurons from the substantia nigra, which are key players in motor function. Surprisingly, the surviving neurons harbor dense protein clusters called Lewy bodies that look like strange black holes. Today, the exact causes surrounding the formation of these Lewy bodies remain mysterious. However, one major player seems to be involved, the endolysosomal system. But what is it and how does it participate in the formation of these Lewy bodies? To understand the infinitely small, it is sometimes easier to use the infinitely large. Thus, in this galaxy, from a dystopian future where humanity is forced to leave a planet out of control, mixing COVID-19 pandemic and global warming, humans must manage the resources necessary for their survival in space, representing here the whole cell. In this galaxy, they organize the various delivery missions from their space station in orbit of the Earth, the Golgi apparatus. And from this station, they send a multitude of spatial ships fulfilling very specific roles. Transport ships in charge of moving materials over a short period of distance, which are called endosomes. Recycling ships, whose mission is to degrade used materials, which are called the lysosomes. Intergalactic travel ships that carry cargoes between several galaxies, these are the exosomes. And finally, energy shuttles that carry the essential batteries to the proper functioning of human constructions, and these are called mitochondria. With this well-functioning system, humans thought they were heading for a prosperous future. However, very quickly, Lewy bodies or black holes appear in the galaxy. From their control room, humans noticed that certain types of shuttles, notably those for transport and degradation, do not arrive at their destination. Worse, they are mostly located inside these black holes. Surprisingly, all the ships do not seem to react in the same way. Indeed, the energy shuttles accumulate in the orbit of the black holes, while the travel shuttles carry their cargo through the different galaxies without ever being absorbed. What worries humans even more is that as these ships disappear, the black holes grow and get inexorably closer to the last bastion of the humanity, threatening their survival. The purpose of my thesis, as you will have understood, is to try to bring a little help to these humans are trying to find a reason why some shuttle types get swallowed by these black holes and not the others. By understanding this interaction, we could prevent them from being absorbed by these sinister stars, which could then stop growing, stopping their destructive progression.
Thank you, Maxime. And this concludes our regular Pitch Talk session 1.